Mesdames et messieurs, bonjour. Bonjour, bienvenue à ce webinaire du pôle IAR. Webinaire qui va être focalisé sur 3 adhérents du pôle, 3 adhérents actifs dans le domaine du biocontrôle. Je vais progressivement passer à l'anglais car nous accueillons Willan Rachel de chez Evologic, qui est une société autrichienne, et Willan ne parlant pas le français, donc par sympathie et correction envers lui, nous ferons la plupart des présentations en anglais. Ladies and gentlemen, thank you for coming. I'm switching now in English. The focus of this webinar will be on biocontrol, and we are very happy to welcome 3 IR members that are active in the biocontrol technology. The first one to present today will be Alex Russo from ERD. ERD, it's a private R&D center specialized in scaling up processes in order to accelerate the industrialization of technology to valorize the biomass. And today, Alex is going to describe their activities in the field of the biocontrol. Alex, the floor is yours. Welcome, and please start your presentation. You can share your screen with us. Thank you.
Thank you very much for the introduction and to invite me to present our activity in BioInput project. So good morning, everybody. So just a few minutes to share my screen. So if it's okay with my
it's
screen.
Okay, you can go ahead.
Okay, thank you very much. So I am going to present you our BioInput activity at IRD. So first, how does IRD implement the BioInput project? So for us, in our project, it's important to develop in parallel the production, the process production, and also in parallel the agronomic trials. So during all the BioInput project, we will test different technology to implement the production of the strain, and in parallel we test our strain in agronomic trials. So after around 4 or 5 years, we obtain for each project a formulated strain active in plot field experiments, and in parallel a production process at a MicroPilot pilot scale. So the first step is to select strains. So we sample soil, crop, or also leaf or plant debris in field. After that, we extract the different strains from the samples, and we screen these strains in vitro trials on Petri dishes, for example. After that, on the selected strains, we made molecular identification to remove the pathogens for human and also for crops. After this first phase, we select around 20-30 strains to go through the second phase. So in this phase 2, we develop a laboratory production process to grow the strain and to stabilize this different strain. And in parallel, we make some trials under control condition in phytotronic room or in greenhouse. It depends on the topic and on the crop. And after this second phase, we select around 5-10 strains in function of the production result and also agronomic result. We want to select the strain not only on the agronomic trials, but on also depending of the production result, to be sure that this strain will be economically speaking good for the market. In the third phase, we scale up the process from lab to micropilot or pilot scale. And in parallel, we make trial in field in small plot, and we can repeat this phase as soon as we have sufficient results to complete the regulatory field. So more in details, the agronomic evaluation. So we implement the agronomic evaluation at different levels. First, at in vitro trials at lab with different tests in petri dishes. Here we can you can see some example of selection for biofertilizer trials. Here it's a Biocontrôle project. And also we see and we look at the mode of action of the strain since the laboratory trials. After that, we go through the trials under control condition in phytotronic room. So in R&D, we have 2 phytotronic rooms and also a greenhouse. So it depends on the crop and of the topic. We can make in parallel trials in phytotronic room and greenhouse. And after that, we go through trials in field with our cooperative on cereal and on sugar beets to select the better strain. During all the agronomic evaluation, we think about the product, the active substance, and also the formulation. So we determine the shape of the active substance, the stabilization, and also the formulation, depending of the strain, but also depending of the application of the product. We think about the type of application, foliar spray, soil spray, or also seed coating, depending on the topic and on the crop. And we see and we look at the crop management technique, the dose of the product, also the number of treatment, the alternance between the conventional product and the bioinput product, and also the phenological state of the crop for the application of the bioinputs. For the production process, so to develop and to make a product with a strain, we have 3 main steps. So first, the liquid fermentation. So IRD is specialized on liquid fermentation only and not solid fermentation. So this first step consists in develop and grow the strain with adapted medium of culture for the culture with different conditions, fed batch, and so on. After that, it's the DSP. The second step is the DSP, with first the separation of the strain from the culture medium with centrifugation or ultrafiltration. It depends on the strain. After that, we obtain a cream of microorganisms. And then we stabilize this microorganism with protective agents. And after that, we shape the product, so we can apply drying, seed coating, also stay with liquid form. And the third step of the process is the formulation. So the active substance is also formulated to have a good application. The formulation depends on the application, if it's a soil spray, if it's a leaf spray or a seed coating. Also, we use green adjuvant, green surfactant produced by Witoleo. Witoleo is in R&D and produce green surfactant for cosmetic, for personal care, and also for conventional product for field. So to carry out bioinput activity, IRD combine 4 main expertise. First in microbiology to characterize and identify the different microorganisms. Also an expertise on agronomy to implement the agronomic evaluation from petri dishes to field. To develop also the treatment strategy and the method of application in an appropriate technical itinerary. The third expertise of IRD is the industrial biotechnology. IRD is specialized in fermentation and DSP process and can design, scale up, and also make some production. And the last is the green chemistry. So we have the experience to design, scale up, and to produce bio-based surfactant and formulation. So now some result on our bioinput project. So first, IRD work on bioinputs for more than 10 years. We work only on microorganism bioinputs and also on their formulation. So we have different topics, biocontrol, with mainly work on fusariosis and septoria on wheat. Also, we work on biostimulants, mainly in water stress. Also, we have topic in biofertilizer on phosphorus solubilization, but now also in nitrogen fixation. And for all this topic, we make a green formulation to formulate all our active substance. Our project are co-funding by Grain and Sugar Beet Cooperative, CRD and Cristallignon. CRD is a group of French cereal cooperative like Vivesia, EMC2, and Cristallignon. It's a big group of sugar beet cooperative to make food sugar. We have also the support of the Grand Est region with a region program. So I'm going to present you some results on two main projects. The first is a biofertilizer project with the target to isolate a bacteria allowed to solubilize phosphorus. And the second, it's a biocontrol topic with a target to develop a biocontrol agent against wheat fusariosis and septoria. So first, on the biofertilizer project. So here we can see a result on oilseed rape on field experiments. So the results are in yield. Here we have a result with 100% of phosphorus fertilization, and on the right the result without phosphorus fertilization. So you can see here the untreated control, and here the trial with our strain. So as you can see, we can win plus 3.6% of yield with our strain. And here, without phosphorus fertilization, it's plus 7.5% of yield. So we are different strains that increase the phosphorus availability on the soil for the crop, and here for the oilseed rape. So on this project, we make field experiment since 2017, and our process production is at pilot scale. For the biocontrol project, it's also a result in field on wheat. So you can see here the untreated control without treatment for fusariosis. And here on the right, the control treated with conventional plant protection product, the Prozaro, in full dose. And in the middle, the trial with the treatment with our strain, with two treatments, beginning of tearing and end of flowering. So as you can see, we can win plus 17% of yield in front of the untreated control. So this result is based on a microorganism with a lot different mode of action. So space competition, but also mycoparasitism and antibiotics production. And with this strain, we have also a result in septoria and in brown rust. Also, we have observed a decreasing of mycotoxin with this strain. On our bioinput activity, we can offer the development of process on strain to design the different steps, liquid fermentation, DSP, and also formulation. We can make the optimization and scale up of the process, and also the production of small quantities of active substance and on formulation. In parallel, we propose also agronomic evaluation from petri dishes to the field. Our customers are our shareholders, but also private company, academics in partnership, and also we have our own projects. We are open to partnership to develop, scale up your bioinput solution, but also to screen and test potential solution, and also to co-develop new solutions. So thank you very much for your attention. And if you have some questions, I'm available.
Thank you, Alex, for this very clear and very pleasant presentation. There is one question for you. Yes. Which technology are you using to dry the cream of microorganisms?
So we have different technology. It depends of the strain, because some strain are sensible to the temperature, and other less sensitive. So we can have spray drying, but for strain not sensitive to the temperature. But also we test a fluidized bed dryer. And also we can adapt the protective agent to stabilize the strain and to make the strain less sensitive to the temperature.
Okay, I have one personal question as well. you are active in the field, you are active, let's say, in scaling up in fermentation, in DSP, and as well in formulation. What is the most challenging part of the value chain, technological value chain, I would say, in bio input for actually to industrialize the processes or the technology?
For me, it's the stabilization because it depends on the strain, but for a lot of strain, the stabilization is very complicated, and the work is mainly in this step to stabilize the strain, to have a strain active in field. Also, the application is very important, so the formulation. So we at IRD we develop the formulation since the lab trials to be sure that we can see all the effect of the formulation in greenhouse and also in photosonic room before the field, and to optimize before the field the formulation.
Okay, we have another question for you concerning the technology to screen soil samples. Can you tell us a bit more about that technology? Technology to screen soil samples.
Yes. So just for the sample on the field, so we take sample, and after that we extract the strain. So we extract the different strain. It's just a centrifuge at lab with high G-force, and after that we can extract with different with water, and also with water with additional molecule to extract well the different strain.
Okay, perfect. Well, thank you very much, Alex, for this presentation. And I let all the people that are willing to contact ERD for a new project to contact directly Alex. She's ready and looking forward to get exchanges with all the participants. Wieland, welcome to this webinar. Wieland is the CEO of Evologic, an Austrian company that transfer cutting-edge technology from pharmaceutical industry to production processes for bioactive in the agricultural sector. And Wieland, the floor is yours. Welcome, and we look forward to see your presentation.
Thank you very much for the kind introduction. Can everybody, does the screen sharing work?
Yeah. Yes, it works. No.
Great. Yes, so basically we have developed a novel bioreactor design allowing to debottleneck the commercialization of fungal organisms. So as we already got some introduction on the field of biocontrol and biostimulants, I'm also gonna spend a couple of slides on that topic. So basically there are still a lot of people starving worldwide, and there is a certain gap in food production between how much food we produce and how much food we actually need to feed the world. However, it would be now pretty much straightforward. Yeah, you just spread out current technologies to a broader, let's say, user base in order to optimize productivities. However, we are also facing quite some challenges, which would be climate change, which consequently leads to pests coming into new regions, also water scarcity, which can be observed even in Central Europe right now. And then there are the increasingly strict regulations for fertilizers and crop protection agents. The list of banned chemical crop protection agents is quite long. Also, the new Fertilizer Act didn't really make it easier for farmers to maintain current yields. Sorry, So there is a need for the next generation of yield-enhancing agents for agriculture, and it makes absolute sense to look at the biological fields, where you can have biopesticides or biocontrol agents which prevent pathogens from establishing a population or directly kill them. And that can be soil-borne, but also airborne, like directly on the leaves. And then there's also the category of biostimulants, whereas for some biostimulants, especially the microbial ones, it's a little hard to differentiate between where does a biostimulant mode of action actually stop and where does a biocontrol or biopesticide mode of action actually stop. However, in more general terms, biostimulants conventionally help to access nutrients with a higher efficiency, basically, or they produce plant hormones so that the plant grows basically better. Altogether, the introduction and commercialization of biopesticides and biostimulants aligns with the UN sustainability goals, zero hunger, responsible resource consumption, also life on land, because I mean the application of chemicals often has detrimental effects on biodiversity, for instance. And if we look at, again, the residues of fertilizer application or chemical crop protection agents, they often tend to either directly go into the groundwater or to persist in the environment and negatively affect, as already mentioned, biodiversity. So, and these biologicals actually promise to overcome these challenges, which is why they're getting a lot of attention, especially in the recent years. So, but these biologicals for agriculture, they have great potential. Everybody knows about it. Like the literature on these compounds is decades old, but the adoption rate seems quite low. Now, the point is that the farmer is basically used to synthetic products like chemicals. However, as I mentioned earlier, they are being increasingly regulated. So the farmer is forced to adopt new solutions. As Thomas also outlined, the development costs of chemicals are quite steep, and the development cycle also takes around a decade, whereas the development cost for biologicals is cheaper. These are numbers originating from average values, and it's not quite clear if industry is just willing to spend more in chemicals because they have the tendency to actually impact the market quite significantly, and biologicals are not, there is not invested as much money in biologicals because there are still some challenges to overcome. However, right now, like the factor is around 1 to 10 of development costs, and around 1 to 3 in terms of life cycle of development. But it's rather clear what the main challenges for these biologicals are. It's shelf life mostly, especially if you look at microbials, but also the application, and I think that holds true for most of them. Generally, biological molecules have the characteristics that they are turned over quite quickly by other biological organisms, and that's a positive trait compared to chemicals, but also for the farmer quite a burden. And in addition, because they're quite challenging to produce, often you have to apply them several times in a season, again, because the half-life, let's say, of a given compound or a microbial is not that high. The associated costs of application are also quite substantial. So here in this little table, I've tried to graphically compare the different characteristics, which are definitely decisive for the farmer, let's say, in the course of the decision which product to employ. So the synthetic products have an incredible concentration. Mostly you have to apply very little per hectare, whereas if you now look at a fungal product originating, let's say, from state-of-the-art solid-state fermentation, you have to apply, excuse my language, but shitload of product for a long period of time, and you have to reapply it. So it comes with a lot of stress for the farmer. That also is then highlighted in the second row here. The application forms the application as such is hardly compatible with other applications. Like you can combine most chemicals with other chemicals without interfering or completely quenching the mode of action of the other compound. But with the biologicals, it's a little tricky. For instance, you want to apply a fungal product as insecticide, but you also have to protect your crop against pathogenic fungi. So you apply a chemical fungicide. So you already see that there's going to be quite some trouble. Also, the benefit per costs. Synthetic products, chemicals are really cheap in production, and most of the time the benefits are quite pronounced. And also here, state-of-the-art production falls behind. Especially hindering because actually there is a fast track in Europe as well as in the US to get market approval. However, current production processes, especially of these fungal organisms, they tend to yield a product which is not completely defined. So you always have some background of other organisms in the product, so you don't have a real specific identity of your product, which is greatly hindering the regulatory approval. That also hinders, like the contamination hinders the reproducibility. And lastly, the stability, which initially only seems a problem for the producer, like you have to deliver the necessary concentration to the end customer so that actually your product performs on the field, is also a regulatory problem because you have to tell regulatory authorities what your effective dosage is. And if within the testing period you're or like in the life cycle of the product during a year, your product concentration decreases too steeply, you run into trouble defining the final product concentration for the regulatory authorities. However, there's great potential in these, especially fungal kingdom. We're looking at something between 2 to 4 million of species, but up to date, only 70% of them can be produced with solid state fermentation, which I've just compared to the synthetic products, but I'm going to go more into detail. So basically summarizing, the product characteristics are somewhat unfit. And that has a very simple reason, that is compared to, let's say, bacteria, these fungal organisms produce mycelium. So it's a relatively large network, and that's quite important for differentiation and spore production. And that is basically the biggest challenge for production approaches. And that basically also makes the production technology key for commercialization, because as much as you can steer and control the production process, as much you can actually tweak your production product characteristics in order to fit the market demand. So I mentioned this liquid fermentation approach with a steered tank reactor just for the sake of completeness here. It actually features high mass transfer, good sterility, and good reproducibility. However, there are only 3 to 4 strains which actually can withstand this type of fermentation, since in a liquid fermentation you have involves steer, and this steer continuously fragments your mycelium, and thereby let's say the natural reproduction cycle of the fungi is heavily impacted, and only the species which are able to form a pellet and survive in pellet form can actually be produced using liquid fermentation. The alternative and more widely used one is actually solid state fermentation. And so basically you grow your fungi or your fungus on a solid substrate that can be inert, so that you have a biofilm on inert carrier material, or you're using rice or cereals as not only carrier material, but also a substrate. There are various designs available, numerous different bioreactor types, but they all have pretty much in common that your mass transfer is pretty much limited, because the solids tend to impair diffusion throughout the bioreactor. So you have a limitation in mass transfer, but also in heat transfer. So actually cooling is quite a challenge in most large-scale production approaches for fungi. based on solid state fermentation. Beyond this fundamental challenges in terms of mass and heat transfer, you come across numerous contaminations, because solid material can hardly be, if you look at rice or cereals, it's quite hard to sterilize them, to sterilize this material on a large scale. And that, of course, impacts product quality and product identity. This carrier material, since you have to use it in order to give the fungi the opportunity to actually produce a mycelium, makes pretty much the carrier material inseparable from your product. So you always end up with a bulk material, because isolating single spores from, let's say, rice is technically and economically quite unfeasible. And also the consistency, I called it reproducibility before, is also quite impaired because of the mass transfer problems. You end up with inhomogeneities in bioreactors, and you hardly can measure anything in real time beyond, let's say, off gas. And that also puts quite some strains or constraints, let's say, on process optimization and process development. So if you look at the value chain here, usually you have product candidates, funnel them into production, solid state fermentation, end up with a bulk material. And given the characteristics of this bulk material, so you have either as carrier material, rice, or inert carrier material, and that basically just allows in for application. But since there are numerous different types of application, that is quite constraining also the rate of adoption. Now, we have come up with an alternative to this. We call it pseudo-solid state fermentation. Here on the left side, you can see the challenges I've been referring to before: consistency, carrier material, and contamination. The features of our bioreactor system, and this is not, let's say, a detailed representation of our bioreactor design. It rather looks like a steer tank reactor, and I'm fully aware of this. However, what I cannot do is show you in a public presentation the full scale technology. However, I can talk about the features. So basically, we have a fully sterilizable vessel and a fully sterilizable carrier material, and that enables us to control the full production process and avoid contamination from inoculation until harvest. Then, regarding the carrier material, we are using biopolymeric particles, which are evenly distributed within the bioreactor to basically protect the mycelium from shear stress. Because we also have in the bioreactor a liquid as surrounding media that allows us to circumvent inhomogeneities while still minimizing shear. So there's only a quite slow turnover of liquids in the bioreactor. Regarding consistency, since we can uniformly distribute the growth matrix across the bioreactor volume, we can maximize the space-time yield. And since we have this liquid, we can measure basically everything within the bioreactor supernatant, and that allows us to estimate and model metabolic activities and use this information to maximize productivity. So basically, the benefits of our system is that we can clearly maintain the product identity. We are quite flexible in formulation and application because we can circumvent the carrier material, like the solid carrier material. We observe high growth rates in our system. We're highly reproducible, and given to our optimization degrees of freedoms, we can achieve a high benefit to cost ratio. So coming back to this table, now this was the conventional solid state fermentation in the middle, and our bioreactor approach allows a way lower application rate because we have a higher concentrated product. Given the avoidance of a solid carrier material, we're way more flexible in terms of application forms. Given our degrees of freedom for optimization, we achieve higher space-time yields and therefore a greater benefit to cost ratio. Since we can avoid contaminations, we have a fully characterized product, and that really eases the regulatory approval process And again, since we avoid this contaminations, the processes also run at a higher rate of reproducibility. And last but not least, given our degrees of freedom in pre-formulation and post-formulation, we obtain a higher level of stability of the final product. And now coming back to the value chain, basically exchanging the method of production here leads not to a bulk product, but to a concentrated powder. And given the low particle diameter, we can basically introduce this powder in irrigation, spray application, seed application, but also fur application, and therefore we have a larger degree of flexibility. Our motivation and offer, I want to briefly outline our business model. So basically we're using our core technology to produce microbials, mainly fungal organisms, which we source from third parties. So we are not in any way active in strain identification or sourcing. And then we distribute, or we pass on the product to our distributors, which are our investors in the seed industry. But that is an optional step and not necessary for collaboration projects. So this is actually the approach for our in-house products. That would be our vascular mycorrhizal fungi. And currently we're also working on a herbicide as a partial glyphosate replacement based also on a fungi. And so basically these products are then dedicated for distribution via investors, and they distribute the product at mostly actually a seed application directly to the farmers. So our motivation or the interests of us of Evologic is generally advancing biological solutions for agriculture. Personally, I'm quite astonished how far behind I personally would deem the European market compared to the rest of the world, since we have the highest regulations possible, like worldwide. Nevertheless, the product adoption rate seems still relatively low. So we mainly focus on non-GMO microbials because, again, if the application is in fields, you should avoid GMOs, especially in Europe. But we also work on plant extracts, since our bioreactor design actually originates from a bioreactor design dedicated for hairy reproduction. But as I already mentioned, we're not doing any screening or strain evaluation activities. The modes for collaboration are basically process development for challenging organisms with ambitious goals. So we conventionally try not to do the standard stuff. A lot of companies are out there willing to do that. So we're focusing on fungal products, but we're also working on bacterial strains. So right now we have started a project on rhizobia, which sounds really boring, but actually there's a lot of optimization potential. Other mode would be starting with a product lead, so collaborative or cooperative approach, including process development and upscaling, or the more conventional contract manufacturing up on technology transfer, or alternatively in combination with a contract development project as a precursor. So putting this into the value chain again, the strain in our case, the majority of the cases comes from outside, or we license it for an in-house product. We take care of production and formulation. We can also go into application development, but we prefer working there with externals because we cannot cover all the bases. And for the distribution, we're flexible. We can offer access to our distribution channels as we have quite substantial investors on board. But we are happy to supply the product just to you. Yeah. Our offer is basically our upstream processing capacity. We have quite a number of fully equipped bioreactors, so small scale around 24. I would call them pilot scale around 5, and production scale 3 × 500 and 1,000 liter bioreactor. We also are involved in downstream processing for the isolation and formulation, which is necessary. I don't want to call it evil, but it's a necessary task because it's just unfeasible to produce a product but not to formulate it. And we're quite actually originates from a pharma background, that we invested quite heavily in analytics. So we are using a mass spectrometry for off-gas analysis, and also do sugar analysis or ion analysis in-house, or qPCR for viable organisms, or quantification of viability, and are also involved in microscopy and large field imaging, especially in context of fungi. That's quite helpful if you want to get a hold of reliable product characteristics. So in a nutshell, we have a patented bioreactor design for fungi and for the production of plant-based biomass. The main benefits are the higher degrees of freedom for application, shelf life, and higher purity. We can offer facilities up to 1 cubic meter, including downstream capacity. We're currently a team of 12 and operating our own independent lab. So we are looking for collaboration partners, potentially as a cooperative project, as a CDO or a CMO project. Thank you very much for your time, and I'm looking forward to the discussion.
Thank you, Wieland, for this very nice presentation about Evologic. I have one question for you. What would be the ideal size for the for your reactor? Let's say adapt as well to the market size of that you are addressing. Commercial scale, I would say, yeah.
It heavily depends on the product and the final concentrations you reach in the production process. So that is really hard to generalize. However, we have found that, I mean, for bacterial products, 1,000 liters can already be quite a lot, especially since the production processes don't take long. For filamentous fungi, we consider the cubic meter scale still pre-industrial. So we are considering, or we're actually planning to build a 10 cubic meter facility next year. Yeah, just to give you an order of magnitude. But again, that really depends on growth rates, space-time yields, and yeah, also the concentration you actually have to bring to the field per hectare.
Okay, and what is again the same question that I passed to ARD at the beginning? What is the most challenging part in your production process for you with this kind of new bioreactor, I would say?
Basically, it's well, actually, our bioreactor helps us to tackle the formulation challenge with more flexibility, especially for fungal products, since the carrier material in conventional solid state fermentation always kind of stands in the way of formulating the product in the way you actually would like to, since you cannot get rid of the carrier material. In our case, our biopolymeric pseudo-solid state is actually part of the final formulation. But since it's like largely water, it's only a low percentage of the final product, and therefore we can adopt basically any recipe necessary. But if you want to go to, or if you're asking for what is the most delicate part, I would say basically the point of harvest and the drying procedure.
Okay, and what is the benefits of having, let's say, a powder microorganism toward a liquid form for a microorganism at the end?
You mean in between what the
at the end, you said that you come up, let's say, with a powder microorganism in one of your slides. And the question I have online is, what do you have the same beneficial metabolic profile with the powder as the liquid form, or is there any difference in activity at the end?
So for us, it was a question of shelf life, and since shelf life are generally metabolic activity mostly driven by active water, we took a rather fundamental decision to pursue dry formulations. because it eases the shipping, it eases the let's say for application. Since as I outlined, our value chain stops at a pre-formulation. So we say, or our responsibility in the value chain stops with the pre-formulation. So we have to reach a point of stability where we can pass on the product to a third party in course of a collaboration, which then does the final formulation for a specified application technology. And that can then be liquid, or it can still be dry. However, we have mostly been asked for dry formulations, and that's why we have decided to pursue that. Sorry.
Okay, perfect. I think that's all the question we have for the moment. There is no more question, and it is 12:15 past 12:15 now. Then probably time to thanks all the. Maybe I will ask Thomas and Alex to join back the floor, to open their camera and their micro, to thank them for this participation. Maybe you have each other a question for each other, because you could not reach the Q&A as a speaker. You have no access to that function. Do you have each other a question for to pass for the presentation, or was it clear for everyone? I know that you know each other. You already have some partnerships and some co-development project as well. Then there is a last question that just appeared now. No, thank you. Sorry.
No, for William, we can talk later, but wondering how's your fundraising going? I mean, as all startup, we always raise funds, but I think that's a separate discussion we can have another time.
Yeah, perfect.
Just want to say hi.
Okay, then I would like to thank you everyone for having participated to this webinar. Merci à tous d'avoir participé à ce webinaire. Thank you, William, for having playing the game from Austria. Sorry, because at the beginning we started a little bit in French, and we probably have a strong French accent. Hopefully you could understand everything what we said. Well, Thomas speaks very well, that's no problem for him, but he used to speak a lot of English actually. You know, when you need to fundraise, to make fundraising, you need to speak English.
Exactly.
Definitively. Although I know that he has a good French shareholder now in his portfolio.
If I may say, thank you very much for thank you very much for doing this in English. I really appreciated it. I'm really happy to be able to participate in your Vivid cluster, let's say, since I'm not aware of a similar community anywhere else in Europe. So I'm happy that you let me join and actually make this work. Thank you very much.
Okay, thank you for this very kind word. I think we cannot. There is nothing else to add. You said everything, William. Thank you very much. Just a last question, last info for the participant and for you as a speaker as well. The webinar will be online in few days, maybe 1 or 2 weeks time to get back. Let's say the full copy of this webinar. It will be on our YouTube channel, and if you want to share with some of your customer, participant, or partners, you're welcome to do it. And of course, if you want us to cut something as well, feel free to do it. We will ask you as well the permission to share the presentation with the participants. If you want to modify it, do it, and just send us, let's say, the sharing version that we can share with all the participants. Thank you very much. Have a nice lunch, everyone, and we see each other, let's say, at the next occasion where we can write something on physical form. Thank you. Bye bye.
Thanks a lot.
Thank you very much. Bye.